but no pressure. Because we're so packed, we are going to try and be as strict as we can for timing. So there will be 10 minutes for each of these sessions. When Sheena rejoins us, there should hopefully be a bell at 2 minutes to, and then a sign when the time's finished. So I'd appreciate if all speakers this afternoon could do their best to stick to time as much as is possible. That would be great. But our first presentation this afternoon is Mei Yi Lim and Katerina Papa from Heriot-Watt University, who are going to speak about AMPER, agent-based memory prosthesis to encourage reminiscing. Over to you.
Thank you, Tom. So I'm Mei Yi from Heriot-Watt, and this is Katerina from Strathclyde. AMPER is an interdisciplinary project between a team of computer scientists at Heriot-Watt and a team of psychologists from Strathclyde. Additionally, we also have a charity organization, the NHS, and a few dementia centers as our stakeholders. So Katerina will start by introducing you to AMPER, talking about our reminiscence agent and the approach that we have taken to develop the AMPER application. I'll then go into a bit more detail on the application and walk you through an interaction flow. And I'll also briefly mention where we are at the moment and what are our next steps. And I'll pass the stage to Katerina. Thank you.
So as most of you know, in this room, Alzheimer's disease is most commonly marked by memory loss, but it also affects other cognitive abilities such as language, problem solving, and the ability to carry out activities of daily living. Autobiographical memory refers to memory for personal experiences and facts about ourselves, and it allows us to maintain a coherent story throughout our lives. And of course, autobiographical memory decline in Alzheimer's disease can lead to a diminished sense of self, sense of identity, and results in low mood and quality of life. So the AMPER project is focusing on creating an intervention that will improve low mood and quality of life in Alzheimer's. And reminiscence therapy is a program that has exhibited positive results in individuals with dementia. It involves discussing experiences and events from our past. It evokes memories. It stimulates mental activity. Thank you. And it improves well-being and helps with depression. It uses props such as pictures, images, objects to assist with the experience. And it can be done in a one-to-one or a group setting. The aim of reminiscence is to bring back some of those memories that are lost, or to maintain the available pool of autobiographical memories to slow down disease progression in a way. So with AMPER, we're trying to create a personalized reminiscence experience in a digital environment where the process is facilitated by this digital character. And the novelty lies in personalizing the material that we will be using, which will be done in 3 ways. We're going to use generationally relevant material that is taken from the BBC Reminiscence Archive. And we're going to utilize what is called the reminiscence bump, which is the tendency for older adults to recall memories from their early adulthood more than any other life period. We're also going to ask the users to upload their own personal material in the app, and we're going to use themes that are of their own preference. So the AMPER project is using multimedia material such as pictures, audio clips, and video clips. And the role of the digital character is to facilitate and guide the conversation between the participants and their carer or partner or friends, using those prompts in a meaningful way. So it is a carer-assisted intervention, and there is a social element involved. The AMPER project itself is a product of co-design, and we have stakeholders from research groups, from charities, and their carers as well. and they have been involved from the early phases of the project. So for example, the digital character, we needed their help and input in terms of choosing and designing the character. And we're gonna give the Amper app to a small group of individuals with dementia and their carers to try out, so we can include their feedback before we proceed with a full-scale study. And Amper is a technology-driven intervention, and it's capitalizing obviously on the benefits of technology, which is remote participation, remote communication, using engaging multimedia, and the ability to monitor and evaluate frequently. So now I'm going to hand over to Mei Yi, who will give you information about the app.
Thank you. So a little bit more about the app. Research has shown that in order for a virtual character to interact effectively and maintain long-term relationship with users, they need to appear and behave naturally. So through a co-creation workshop with our stakeholders, they've recommended that we should use 3D cartoon older human-like characters. So we selected a bunch of these characters with those criteria and ran a survey. And these are the 4 characters that we ended up with. So in the Amper app, they will be able to select one of these characters to interact with. And in terms of behavior, since reminiscence rely heavily on memory retrieval, our character will have biologically inspired human-like memory where it applies spreading activation for retrieval. So spreading activation is a process where one element reminds you of another. For example, if I mention the Edinburgh Fringe Festival, that might remind you of someone you have been to the Fringe with, and that in turn might remind you of the shows that you have been to and how you felt at that time and so on. So in the same way, the character will first select a story that is of interest to the user. And then based on the user's feedback, whether they like or dislike the story, it will continue selecting related stories or different stories. And this process is really important to ensure coherence and continuation of the reminiscence experience. And all this interaction will take place through an Android tablet. Here's a snapshot of the tablet interface. You can see that the character is on the left side of the screen, and on the right side, there's a carousel of cues of which the user will be able to slide through. And for each of these cues, the character will be providing a short description and provide a prompts to encourage discussion between the user and their carer. And on this screen, I hope you can see there are 2 buttons which say do not reminisce this cue and do not reminisce this story again buttons in red. These 2 buttons are really important to enable the user to eliminate a particular cue or a whole story altogether from future reminiscence in case it's something that upsets them, because we do not want any distress during this reminiscence process. So now a walk through the interaction. The character will always start by greeting the user. And if this is the first time they are interacting, we'll be collecting, they will need to register and we'll be collecting information about the user, such as their age and interest, because these are the factors that we're using for retrieval. And we'll be giving a tablet to each participant, and only that participant will be able to reminisce using that tablet. So if this is not the first time they're interacting, they will be asked to identify, to confirm their identity. If they are not the respective person, they will not be able to continue interaction. And if they are, then they will be asked if a carer is present, because as we mentioned before, it's carer-assisted reminiscence. If a carer is not present, they will be shown a slideshow where they will be able to view all the previously reminisced cue. If a carer is present, they will be presented with 3 new stories of which they can reminisce. They can choose to reminisce only 1 story or all 3 stories. And when they are done, they will be asked to provide feedback. And here's a short video. I hope it plays. Of the interaction.
Yeah, Billy Connolly. This picture shows Scottish comedian Billy Connolly. Have you ever seen a comedy special?
Yeah, definitely. You went to see Billy Connolly?
Aye, we went to see a show in Usher Hall.
So yeah, that's just to give you an idea of how the interaction looks like. And so at the moment, we are almost ready for our first pilot study with healthy adults where we evaluate the robustness and the usability of the system. And once feedback has been gathered and refinement has been made, we hope to move to the next phase of the study with our target group. And we would like at least 40 individuals in total. And in the first phase, we'll be looking at personalized versus non-personalized version. So in the personalized version, as Katrina mentioned before, we'll be taking into consideration the user's age, retrieving materials from reminiscence bumps, and then their interests, as well as personal material presentation. And in the non-personalized version, story will be selected randomly from BBC RAM. In the next phase, we hope to look into adaptation where the system doesn't only personalize the content, but also adapt to the changing needs of the individual. For example, if they realize that their attention span is reducing, they might be presenting less story or less cues. And that's briefly about AMPA. Any questions?
Thank you very much. And that's an excellent reminder to me that I forgot to mention at the start, that just a reminder that if you've got questions, if you could put them on Slido, that will help us to collate them and hopefully ask them when the time is available. I'll make the foolhardy suggestion that if there are any questions we don't cover, we will do our best to try and get answers to and circulate after the event to the best of our ability. That's often easier said than done. I would now like to invite Conor Dolby from the University of Glasgow up to speak about his project Deep Thickness, a novel deep learning method for estimating cortical thickness trajectories in Alzheimer's patients and healthy populations. That's a mouthful, isn't it? But I think Conor is a really good example of the coming together that this conference symbolises, because before doing the PhD, Conor was one of the clinical studies officers with us in NHS Lothian with the Neuroprogressive and Dementia Network, and is sorely missed as part of the team, but still does some techie things for us, which we're very grateful for. But it's this kind of shifting between clinical research and university research that is exactly what we're trying to epitomise in the coming together of Scotland. So over to you, Conor.
Thank you. Hello, my name is Conor Dolby, and today I'll be talking about my PhD research project. As he's already read the title, I won't read it again. With no disease-modifying treatments available and several challenges in diagnostics, there is a call for new innovations in AD research. Interestingly, research suggests that cortical changes can occur up to 20 years before the onset of clinical symptoms. And one of these changes may be in cortical thickness. Cortical thickness can be defined as the distance between the white matter and the pale surface. And several authors have already documented how cortical thickness can change over lifespan. Researchers also linked cortical thickness as a signature of Alzheimer's disease. We intend to explore this further and link cortical thickness as a potential biomarker for Alzheimer's disease. Specifically, we aim to generate a normative model of cortical thickness for cognitive normal, Alzheimer's disease, and prodromal mild cognitive impairment patients. From this model, we can not only identify and classify unknown individuals into their clinical groups, but we can also predict disease progression using clinically meaningful metrics such as cognitive decline, loss of independence and functionality, as well as even predicting when they'll enter the care system. By measuring cortical thickness, we aim to detect the presence of disease significantly earlier than the onset of clinical symptoms, thus offering us an opportunity to intervene before the pathology can progress to a stage where actually successful intervention is quite challenging. Now, to generate this kind of normative model and make subject-level inferences, we need a large data set and a large collection of data sets with diverse populations. An example of this might be the UK Biobank, which contains 60,000 volumes. Now, if we wanted to measure cortical thickness on this data set, we could use existing methods such as CAT12 or FreeSurfer. However, these are associated with extremely long computational times, with 7 hours per volume, which, when you scale it up to the biobank, that would take approximately 48 years using a single computer. Moreover, even when we use FreeSurfer, which is considered the industry standard, it still makes several critical errors, especially when using clinical data, as exemplified by these 3 different data sets, whereby the yellow arrow indicates where FreeSurfer has failed to identify either the white matter or the pile surface, and cortical thickness cannot be estimated. Finally, researchers identified systematic differences between these methods, questioning the validity and reliability of these methods. To account for these limitations, we have leveraged recent advances in artificial intelligence and deep learning to create our model, Deep Thickness. Now, Deep Thickness takes an individual's T1 weighted MRI volume and outputs cortical thickness. And unlike FreeSurfer, which takes 7 hours, we generate cortical thickness in a mere 35 seconds. Apply that back to the data set of the biobank for 600,000 volumes. Instead of 48 years, we can process that in 24 days. Now, we also, as part of our model, produce a 3D reconstruction of that individual's cortex, and it perfectly aligns with their MRI volume. We overlay cortical thickness estimates as a mesh color temperature overlay, whereby blue is cortical thin parts and red is more thicker parts of the brain. Notably, we can also generate a subject-specific reconstruction of the white matter as well. Now, to exemplify how we could use this model and cortical thickness in general in sort of Alzheimer's disease context, also just to highlight that we actually improve our accuracy compared to FreeSurfer. As you can see, we have far less yellow arrows compared to the industry standard of FreeSurfer. As I was saying, to sort of illustrate how we can use our model and cortical thickness for Alzheimer's disease, we took the Alzheimer's Disease Neuroimaging Initiative dataset, which contains about 3,600 MRI images from 400 participants across longitudinal visits for about 9 years. And here are the results. First, we see a distributional shift between the clinical groups. However, the distinction is not that great. But when we actually plot this across age of the participants, we see a greater distinction, as you can see here. Now, this distinction and delineation between groups is even further emphasized when we plot across time, whereby in this graph, 0 months represents the baseline visit, and then we, in the study, they measured across multiple visits across sort of 9 years, and you can see a clear distinction. Now, we can not only measure cortical thickness of the whole brain, but we can actually do this regionally in areas that are vulnerable for Alzheimer's disease, such as the entorhinal cortex. Here again, you can see a similar trend and delineation between the groups. This is also reflected in other important areas, such as the temporal region for things like memory, the parietal region for motor and sensory, the frontal region for decision-making, and finally, the occipital region. Now, when we finally delve a bit deeper into the analysis, when we look at how cortical thickness changes across time, we can see that this trend and delineation is also reflected in cognition, as measured by the MMSE Mini Mental Status Exam score. And hopefully you can see how similar these sort of trajectories are between our cortical thickness estimates and the participants' cognition. And we hope that these preliminary initial findings can exemplify the potential benefits of using cortical thickness for Alzheimer's disease. Going forwards, we hope to amass a much larger collection of data sets with more diversity in the populations. We're also looking into using the regional changes in cortical thickness and how they compare with cognitive domain scores, such as your memory score on a cognitive test with your temporal region, or perhaps your visual spatial score in relation to your occipital lobe. We're also going to extend cortical thickness as a measure for other diseases, such as MS, Parkinson's, ALS, stroke, and we're actively looking for collaboration with researchers and clinicians for those sorts of projects. We're going to validate our cortical thickness estimations using qualitative and quantitative measures. And finally, we're going to further refine the mesh topography using graph neural networks and sort of account for any remaining errors in that segmentation of white matter and pile matter. I'm happy to go through any of these in questions, or if you want to come chat to me afterwards. But otherwise, I'd like to thank my collaborators, thank the MND for inviting me, and thank you all for listening. Accept any questions. Thank you.
Thank you very much, Connor, and thank you very much for keeping to time. So hopefully we'll be accumulating some questions on Slido, and if we continue as we're doing, there'll be time for them after we've heard from everybody. It's now a great pleasure to introduce Lucy Sterland from the University of Edinburgh, who is going to be speaking about dementia alongside other conditions, an upcoming priority setting project. Over to you, Lucy.
Hi, everyone. Nice to see you all. Thanks very much for all coming today and for having me to speak. So I'm Lucy Sterland. I'm an old age psychiatrist in Edinburgh with NHS Lothian, and also a researcher at the University of Edinburgh and an Atlantic Fellow for Equity in Brain Health with the Global Brain Health Institute. The Global Brain Health Institute has funded a project that I'm about to start doing this year, and that's what I'm going to talk to you about today. So my research interest is the interaction between having multiple conditions, long-term conditions, and dementia and other mental and brain health problems. Now, multimorbidity is a name that's sometimes used for the coexistence of two or more conditions, and this has been identified as a really important area for research and policy, particularly over the last 10 to 20 years. And this was exemplified by the Academy of Medical Sciences report in 2018, identifying multimorbidity as a priority for global health research. And in that report, they also emphasized the importance of including multimorbidity with dementia, because they acknowledged that with aging populations, more people in the world will have multiple conditions and dementia alongside that. And they noticed in this report that this was an area that hasn't had a lot of research or attention, but that is going to become increasingly important and needs more involvement. So why is it important to think about multiple conditions alongside dementia? Well, I think the clinicians in the room won't be surprised to hear that this is something that is really important in the life of people with dementia, that we often notice that dementia research focuses on the brain, and brain changes possibly to do with proteins or imaging, particular specific types of what's happening in the brain. But for somebody living with dementia, they might have other things happening to them which might have more impact on their daily lives. So for example, somebody might have a lung disease, they might have arthritis, and they might have problems with digestion. So for all those conditions, that person might be taking medication, and the interaction between the medicines and their ability to think and process information, and then in turn reliably take the medication that treats the physical conditions, might all be affected. So we know that there's a really important interaction between having multiple conditions and having dementia. We also know that some people who have multiple conditions have cognitive impairment, so difficulty with memory and thinking, but that isn't diagnosed as dementia because they have so many other things going on that it's considered not to fit the pattern of a typical dementia. We also know that from research, people who have multiple conditions alongside dementia have an increased rate of attendance at hospital and GP practices, which obviously is costly to services like the NHS. They also have faster mortality, so increased rates of dying more sooner, and then lower quality of life overall on average. And I think probably wouldn't surprise you to know that people with dementia have, the majority of people with dementia have at least two other conditions. So this is research from a GP-wide study of about 1.2 million people in Scotland, looking at GP records. And the researchers had a list of 40 conditions, and they took all the conditions and saw how many people had each condition. And they found that of people with dementia, only 5% had only dementia. 13% had dementia and one other condition, 18% had dementia and two other conditions, and 64% had dementia and at least 3 other conditions. So this really is the norm for most people with dementia. But unfortunately, a lot of research excludes people who have multiple conditions, and that's not exclusive to dementia. Lots of other, particularly drug trials, will exclude people with other conditions because they need to focus on one particular thing that's happening. So this brings us on to what I'm looking at in this upcoming project. So I'm really interested in the outcome of dementia trials, so the drug trials that are being done and other studies that we're looking at, and what sort of things are being studied, what are the endpoints, what are people targeting with treatments. And most commonly in dementia trials over the last 10, 20 years, these have been cognition, so memory and thinking, unsurprisingly, but also things like function, so ability to carry out activities of daily living, quality of life, symptoms such as mood and behavioral symptoms, and then also more recently, as we've seen, the clearance of amyloid protein in the brain. And my question really for this upcoming project is, are these outcomes that are already existing in trials and other studies, are they important and meaningful to people who have dementia alongside other conditions? And there is some research already about what matters to people with dementia. So particularly from the angle of patient and public involvement, like that we heard from Rosie earlier on. But some of those studies are largely about researchers and what researchers think is important to people with dementia. There are some studies that have included people with dementia. So there's just a few here just to demonstrate that there are a few studies looking at outcomes important to people with dementia. And then there's one project that's looked at what priorities for research people with multiple conditions have identified. But what I'm interested in is the overlap between this, and I don't think we have any evidence about this at the moment. So the aims of my study are going to be to identify the outcomes that are meaningful to people, and then to co-produce with the people in the group a list of person-centered priorities for research. And the aim for that will be that it'll inform my own research going forward, but we'll also publish it to the wider research community and policy to make that available. So we're going to hold some focus groups. These are going to take place on Teams online, because although we know that has some limitations, we know that it has the benefit of meaning people can access from a lot of different geographical backgrounds. And we're going to include people with dementia or cognitive impairment who have at least 2 other conditions. And those people will also be invited to bring a supporter if they need to help interact in those sessions. And then we're going to have a parallel group for people who've been carers or care partners for people with dementia and multiple conditions. And there'll be 2 facilitators, which will be me and Rosie, you've met already. And what's going to happen is we'll have 4 parallel focus groups. One group will be the caregivers, and the other group will be people with dementia. The first 4 sessions will happen in parallel, so 2 separate groups, and then we'll come together for the 5th group to share the priorities and make a list together. And then at the end of that, we're going to publish and share the priorities with a launch event. So hopefully this is something that we can all watch this space that's going to happen over the next year or 2. So when we're in the group discussions, we're not going to reinvent the wheel. We're going to look at what's existing already. So there's various different projects that have looked at outcome measures in people with dementia. And this is some work that was commissioned partly through the Scottish Government in 2017, identifying outcomes for dementia trials. And they're writing quite small, but it's things like cognitive symptoms, social engagement, daily living activities, and so on. Our aim will be to look at these and think, okay, we're not starting from scratch here. We've got some information. Which of these do we think are important? Which do we think could be changed? And really, how relevant are these things to people who are managing multiple conditions alongside dementia? I just wanted to touch a little bit on some challenges I faced already before even starting the project. So this project is, I think, largely patient and public involvement work. So it's about finding the priorities of people affected by these conditions. But it has been classed as research. So that means it needs to go through the ethical approval process, which of course is an absolute necessity and brilliant. It has made some difficulties in co-producing the work, because my aim was to have all of our discussion guides for focus groups co-produced with people in the group and people with dementia. But I couldn't get the money to start the project until I got the ethical approval, and the ethics board wanted all the discussion guides to be approved before we started the project through the ethics. So I couldn't get the money to support people to help me co-produce the guides until I got the ethical approval. So I ended up in this sort of slight tangle, which I think is something, when we think about patient and public involvement in research, that there are quite a few challenges still out there about navigating through the processes with ethical review boards and things like that. So I'd just like to invite you to watch this space. We're hoping to start recruiting in August 2024. And if anyone is interested, please contact me on my email address. I'm also available to talk if anyone wants to discuss the Global Brain Health Institute. I've found out recently that their next round of applications is open for the Atlantic Fellowship for Equity in Brain Health, which is a year-long program in Dublin or San Francisco. And I would be happy to chat to anyone who's interested. Thank you.
Thank you very much, Lucy. Great. Now, can I invite Martin Quirk to come up to give the next talk, please, which is Designing Homes for Healthy Cognitive Aging, Lessons from Co-Designing Prototype Homes Using Virtual Reality. Over to you.
Right. I was thinking earlier about these various definitions of early career researcher, and there's different definitions from different places. I think it's to do with whether you're nervous before you come up onto stage. So my heart rate's currently about 30 beats a minute higher than my normal resting heart rate. So let's see how we go with this. So yeah, I'm Dr. Martin Quirk. I'm an architect by background. I'm based at the University of Stirling. So I'm here to talk about a project that was led by Professor Alison Bowles. Quite a lot of parts to it, so I'll give you a brief overview of that and then kind of get into the nitty-gritty of the bit I want to talk about. So stats that you're kind of all be quite familiar with, this idea that people turning 65 will spend half of their remaining life with some sort of health condition. And as of 2022, dementia is the leading cause of death. The vast majority of people would prefer to live at home if they've got a dementia diagnosis, but even just in terms of kind of visibility, in terms of physical impairments, only 10% of our homes at best are able to support the person living at home, never mind have guests, visitors. And what we found in the kind of evidence that's there, I'm getting a feedback loop here, I don't know if others are, that when physical adaptations are made to someone's home, it's far more likely to be about physical issue rather than a cognitive issue. And that sort of maybe kind of, I guess, links to what Lucy was just talking about, that, you know, it's a bit more complicated than just one condition at a time. So stand
a little closer, it might help with the feedback. If you just stand a touch closer.
Right, okay. Yeah, that's slightly better. Now I'm deafening myself. So the project was quite a big project, lots of partner organizations, local authorities, and kind of businesses involved. A whole heap of different activities here that might bring back some good and frustrating memories to some of my colleagues who are here in the audience, that kind of covering all of these activities and how they overlap with each other. But the bit I want to talk about was virtual reality co-design workshops that we undertook. So the idea was to identify scalable solutions that allowed people to, I guess, to live their best life, and staying at home in their own house. We started with 2 kind of prototype home designs, and then through 3 rounds of review, how we had participants using VR headsets, and sometimes on a kind of tablet kind of equivalent of that, review the designs. And each time we'd collect their feedback and then put that back into the process and update the design, and then do the same thing again. There was about an equal mix of people, older people, so varying degrees of cognitive capacity, and then the other half were varying housing professionals, so architects, planners, housing managers, that kind of thing. The first 2 rounds were on the basis of kind of 1-to-1, where this person had the headset on, and then a researcher was kind of facilitating them, kind of guiding them through the process. And in the 3rd round, where it was more of a kind of group discussion, because people could sort of make a comment, and then other people were able to kind of respond to that comment. So the first round might look a little bit like this image here, where the researcher's kind of watching out for the person to make sure they're not bumping into furniture. But then the second round, where we have what they're seeing cast onto a screen, and then others can participate in that. So that meant we could get much more involvement, and we had people who maybe otherwise might have been hesitant to kind of participate, or then feeling much more comfortable when they were in the group. So to get that to play. Okay, so this is essentially what's happening. So you've got the person kind of navigating their way around the space virtually, and then we're prompting them with questions about what they like or don't like, and this is all in the sort of the kind of provide the idea of we're trying to design a home for your future self. So we had difficulty with some of the professionals to get them to switch into talking about themselves rather than for a notional client. Okay, so I've sped through that part, so we've got a bit more time on this. So the kinds of things that we're were kind of coming out. And some of this was confirmative of what we kind of knew from the kind of wider literature around designing for aging, particularly designing for dementia, was the example of the actual layout is really important, so visual access. So you walk in the door, you are able to understand, where's the loo, where's the kitchen, that kind of thing. Or you wake up in the morning and you're not sure maybe why you've woken up, oh, wait, now I need the loo, because you get the prompt of seeing the toilet. Or if you're in bed or in a seated position, that you're able to take advantage of seeing out to the landscapes of the kind of therapeutic value of being able to have that view. So that view didn't have to be, this amazing view of the sea, but that was great if it could be. But even just a smaller scale attractive garden was seen quite valuable. So small details then, like the height of the windowsill, became really important. Ergonomics and safety came up quite a lot because we have people talking about issues to do with tripping. So that might be to do with the change in material from one room to the other. So if you had a change in tone, so a light floor to a dark floor, they would say, okay, I'm very likely to sort of trip up there, or my mum or my dad would have done if they were here. And with the same way with things like furniture, where you might have a sharp corner on a table, let's avoid that. So lots of kind of really practical things coming through. The light and lighting one was the one that kind of surprised us, in that no matter how bright we made the model, the participants were saying it's still, a lot of participants saying it needs to be brighter, needs to be brighter, needs to be brighter. So I guess it ran home the suggestion from the previous evidence is that lighting levels need to be 2 or 3 times higher than for the older population than they tend to be for a younger population. And that's just to be able to sort of see to the same level of detail. And that's really important in where you're doing closer work. So in the kitchen, where you're trying to use a sharp knife to prepare a meal. And and we know all about the sort of issues to do with someone with dementia. If you take away their the opportunity to do these things, they lose the ability to do these things. So it becomes kind of even more important in maintaining maintenance. Right through to storage, which was a really interesting one because we had, again, professionals who sometimes were a lot younger, right through to kind of much older, older people as participants. They were still coming back to the same thing. Storage is really important, and where it's located is really important. So that space just inside the door, again, is this really, really valuable place to kind of put a lot of effort into how you're designing. So the variance across the participants, I guess, then was where it started to become more interesting, because the anticipated lifestyle of people varied quite a lot. There's my time running out already. Okay, I'm very good at going on tangents. Lost my train of thought now. Let's skip right forward. There's a whole heap of things to cover there, which I'll skip over. But what may be valuable then as next step, so I get off the stage and let the next person up, is here's some information where you can go and look at the web viewer of the web version of that VR viewer that we use for the research. And then I'll shut up and leave the stage. Thank you, everyone.
Fantastic. Thank you very much, Martin. I think that's a lesson that a bit of anxiety and an increased heart rate can kind of be associated with good performance. That's we all need a bit of anxiety in our lives. So last but not least, I'd like to invite Jodie Watt up to speak to us for the last presentation in this session, which leads very nicely on to the following talk. But this one is about identifying drugs for repurposing in dementia using the UK Biobank cohort. Over to you, Jodie.
Hi, so I'm Dr. Jodie Watt. I'm a postdoc at the University of Glasgow working with Professor Terry Quinn on drug repurposing for dementia. So where are we with drugs for dementia? As we all know, dementia is a situation of huge socioeconomic burden, and we ultimately lack effective and financially viable medications for all of our patients. We've had certain drugs for a while, I'm sure you've heard of all of them, but the benefits of these are often small. And then we've had newer drugs that are very exciting, but it remains unclear how viable these will be for widespread patient use in highly burdened and overspent healthcare systems. So the question is, can we repurpose drugs that we already use? The great thing about this is that it would save us both time and money, and we would already know about the safety profile of these drugs. So how do we identify suitable drugs then? So biobank. Biobank has already been talked about several times already today, but the great things about biobank for us is that there's a lot of cardiovascular information, there's a lot of drug information, and there's a lot of people. So how do we want to do this? With something called Pheasant, or Phenome Scan Analysis tool, which is a very biobank dedicated tool that is freely available. And if you have any interest in that sort of space, I would encourage you to have a look at it. That's the paper there. So just a quick overview of our materials and methods. We used the complete UK biobank cohort, meaning that we didn't exclude anyone. And we did a phenome-wide association study. We looked at all cardiovascular risk factors that we deemed to be relevant, and all the medications that were reported by individuals. So for some of these, they might be listed with a slightly different name, but still be the same drug, and we did deal with that as we went along. It's hypothesis free. Essentially what I meant by this is we're using the complete biobank cohort again, so we're not excluding people who have like traumatic brain injury or anything. Everyone's going into it and we're just sort of seeing what happens. And our outcome of interest is those who developed all-cause dementia. Analyze using Pheasant, as I said, and we adjust for their age at their first visit, because this is predominantly done in first visit, sex, and then the Townsend Deprivation Index as well. And we have a very specific significance threshold for this work, which essentially we have taken from other similar studies, and they use this threshold. If we don't use this threshold, if we use, say, P equals 0.005 or whatever, we end up with a lot of drugs. But we are looking at that data at the moment, but it's not for this conversation just now. And as I said, we cleaned the data to address where there are repeated drug names and synonyms as well. We looked at the full cohort. We then tried to deal with some of our indication biases by looking at a cardiovascular subcohort, and then we also looked at drug classes as well. So I'll have to speed through this, but essentially Pheasant associates each phenotype with all-cause dementia, and is tested using one of several types of regression using this decision tree, which is available in the paper. Again, we adjusted our models. So when we look at the full UK biobank cohort, so that is literally everyone who had information about age and sex. So there are a few people that drop out if they don't have that information. And this is sort of overview of our cohort. There's just over 500,000 people in it. The vast majority of them currently drink. The majority of them have smoked at some point or another. 5% of them have diabetes. Almost 25% of them have high blood pressure that's been diagnosed by a doctor. So some people might have it, but it's not been diagnosed by a doctor. And then, as mentioned, we have our all-cause dementia group that we're interested in as well. So when we look at our cardiovascular medications, we find our strongest associations with insulin, aspirin, and metformin. So we're talking about diabetes drugs and a pain drug. You will see that they're also all positively associated with dementia development. The problem we have here is the indication biases. Well, we have several, but the main one is the indication bias here. So we wanted to see whether our effect differed within drug class as well. We didn't have a huge amount of drugs to do this with that were significant to our threshold, but with these guys, so the ACE inhibitors that we corrected for blood pressure indication, so these drugs are predominantly used for blood pressure reasons, high blood pressure reasons, they weren't significantly different to each other, but obviously we would like to test this more with an array of drugs that are ACE inhibitors rather than just these two that were significant, but that's work that's ongoing. And then we did a cardiovascular subcohort. So part of our rationale was that if we did a cardiovascular subcohort, everyone in that group would have cardiovascular conditions, and therefore we can start dealing with some of our indication bias. Defining a cardiovascular cohort turns out to be really quite difficult. If we do it on the basis of just ICD codes in Biobank, you don't get anywhere near as many people who actually seem to have cardiovascular conditions. For example, if we just do ICD codes, we get almost 39,000 people. If we pull in the variables diabetes diagnosed by a doctor and vascular/heart problems diagnosed by a doctor, we suddenly have 166,381. So that's the cohort that we went with. Again, most of them currently drink, most of them have smoked at some point, and all of them have cardiovascular conditions. So when we looked at this, essentially we had the same thing again. So our insulin, metformin and aspirin, but these are all the ones that are significant to this threshold that we mentioned. So things like sildenafil, which is Viagra, are also significant as well. And then we did a bit more in trying to deal with our indication bias. So we wanted to look at drugs that have multiple indications and correct for their cardiovascular indication. So we did that here. So for example, if you take sildenafil again, we corrected for blood pressure indications, but we didn't correct for like individuals who might be using it for like erectile dysfunction reasons. And within this, we found that the relationship was still significant for, like, there was still a significant likelihood of having dementia, or a significant relationship with dementia, all-cause dementia, for all of these aside from metformin. And then we also try to do this where there's a singular indication for the drugs. So we did that with, like, say, insulin, where it's diabetes, but all of these guys are significant, which suggests to us that we need to get a further handle on our indication bias as well. And then for some conclusions then, we demonstrated the utility of large-scale studies in elucidating medication associations with all-cause dementia. We need to validate this much further. We've got problems of reverse causation, additive effects, indication bias, and various other things as well. We want to further validate against more sensitive surrogate markers. So, for example, we want to look at the MRI data. Brain Health Index is something that I worked on previously, which combines different types of MRI and might have additional utility in this sort of drug study. And we're collaborating with Cardiff at the moment. So they have access to the SAIL data bank. So instead of 500,000 people, they have, I believe, around 2 million people that they have access to validate what we're doing. They also have, thank you, they also have data on duration on the drug, which is a huge assumption that underlies biobank work. And the work that we're doing is that we don't have that data readily accessible. And then we're going to share our findings with an expert panel to determine how valid they are and which to prioritize in our future work. And then, yeah, so these are just a whole bunch of people to thank for their involvement in the study. And that's me.
That's exactly what I was going to say, Helen. Perfect. And we are, because everybody's stuck to time, we have time for questions, which is perfect. So there may be some happening in the internet somewhere. I don't know, but we've also got people here with mouths. So has anybody got a question that they would like to ask of any of the panel, and we can ask them to respond? Any questions in the room?
We've got one for Martin. This one. We're keen to hear how those with dementia find the sliding doors when closed. Is there challenges to the way they're facing?
Do you want to pop up here? That's probably easiest, Martin, if you could respond.
Sorry.
So there was, I guess with that question, there was a balance between what people were familiar with and where they were seeing a huge advantage in the space saving. So you're trying to, navigate your way around a door that takes up, a normal door will take up a full square meter of your house, whereas the sliding door won't take up any space at all. There was a suggestion that some folk might find some difficulty understanding that it needs to slide, and then maybe it then needs an indicator that with an arrow that tells you that. But they didn't, amongst the cohort that we had, they sort of seemed to find that was far more advantageous than disadvantageous, especially where it was dividing the lounge rooms and dining rooms and those kinds of spaces that you might want to leave open plan more of the time or not.
That's great. Thank you very much indeed. So we didn't really give much of a chance for the room. Is there anybody here who'd like to ask a question about any of the presentations we've just heard? Great.
So my question is for Connor. With regards to the deep thickness model, is it trained in a supervised manner? And if so, where did you get the ground truth from?
It's a great question, and it's a difficult question to answer because we use free surfer as a ground truth, but the benefit of learning and artificial intelligence is that even though free surfer is still making errors and we're calling that our ground truth, we're calling that's what's correct, the way in which deep learning works is it learns an abstraction of what cortical thickness is like from the MRI image, so it can learn beyond the errors that free surfer makes, and that's kind of the beauty of AI and deep learning. I hope that answers your question, but generally there is no ground truth. There's no real ground truth because it's a 3D problem that we're tackling with a 2D plane.
I'm just struck by the overlap between Connor and Pontius Pilate there. What is truth? Any other questions? There must be from the. Oh, there's a Slido question. Excellent.
There are many online questions.
Oh, good. Okay, excellent. We'll make sure we give an opportunity to the group.
But yeah, So we could go back to one for Lucy then. The question from the data you presented, Lucy, can having long-term diabetes pose an increased risk of dementia?
Thank you, and I'm glad I saw that question in advance, so I've had a chance to think about it. Yes, we know that diabetes is a risk factor for dementia. Not that means that everyone with diabetes will get dementia, but it can increase the risk. And I suppose there's a lot of work ongoing at the moment from different types of research, thinking about how and why different conditions and combinations of conditions can increase the risk of dementia and affect people's experience of living with dementia.
Thank you.
Got a question for Jodie.
I was just going to say as well, if anybody does have a question in the room, just stick your hand up and we'll come to you. But otherwise, we'll go with the Slido questions for now.
So for any of those drugs identified as having an adverse impact on dementia or even a contributing factor, have any alternatives been identified?
So I think that's a really good question. I guess that's kind of part of what we hope that we can identify in the longer term. These are the drugs that are just sort of like mentioned and reported by the participants. So it looks at every possible drug going. This is what I meant when I was talking about sort of not having a hypothesis. So yeah, I think the first line is to identify the ones that definitively seem to have a relationship, and ideally we would identify ones that have a negative relationship, i.e. might be protective. That's what we were hoping to identify, but that's not what we've seen to this point. So then take those drugs forward and repurpose those drugs ultimately, which would kind of bypass the need for alternatives in the way that you might have meant, if I've understood the question.
I can see the Daily Express headline already, though, Viagra increasing your risk of dementia. That would be everything the newspaper likes coming together in one article. Oh, there's a question at the back. Excellent.
Oh. Thank you. It's also for Jodie, actually. Did I remember that right, that you also said you used the deprivation index to filter how exactly were you using that? What did you filter in with that?
So we're not filtering. It's just, it's literally like Pheasant goes through all your columns of data. And deprivation index is one of those columns. So using the decision tree that I showed, it pulls deprivation index in because it's in the original data file. And then we also list it as a covariate. When you code into Pheasant, you have an option to pull in a data file that has your covariates, and we just copy that column into it. So there's no like separating by like sort of arbitrary bounds within the deprivation index. It's just pulled in as a continuous variable.
Great, thank you. We've got time for one or two more, and it sounds like there are hundreds.
There are lots, but maybe for the Amber study team, we just put a couple of the questions together, I think, asking about triggering the memories. So how do you know that the prompts and questions will be relevant, and have you had any problems with triggering unpleasant memories? Sorry, and the final one to that was, and then how do you record how the memories are discussed so the individual can access it afterwards? So.
Thank you. So there are three questions there. For the first question, we do not know yet about the triggering of bad memories because we haven't run any study with our target group. And the second question was about triggering memory. So we will be using their age to target materials that are in their reminiscence bump. And then we will also be collecting their interest in the sense that it's which themes in life that they are interested in, for example, sports or artists or music and so on. And then we'll be targeting those materials. And the third question, can you remind me again?
How do you go back to the participants so they can visit it later?
Okay, we will be recording what they have reminisced before, but we will not be recording the conversations between the participants and their carer. So the carer will be providing us with some diary of their interaction about how the participants felt about a certain cues, about what they have discussed before, whether they got distressed or not. And then based on all those, we will, for the future interaction, we will perform the spreading activation and so on. And Katrina has something else to add?
About the first point, there are options within the app that if there is any material that they don't want to interact with for any reason or they're distressed, they can choose not to interact with it anymore. And the fact that the carer is present is to facilitate a pleasant interaction, let's say. So if they are distressed, there will be someone to support them. And the app itself supports that by rejecting material and not being presented with again. But we need to start piloting first just to see if there's going to be something there. Thank you.
That's great. Thank you very much. So maybe we'll draw to a close there. Just for clarity as well, in case anybody's not familiar with the term, the reminiscence bump isn't a return to phrenology and measuring bumps on the brain and the skull, but it's a phenomenon in memory that it seems to be that between the ages of about 15 and 30 is when we retain the most vivid memories. So for me, that's the mid-90s onwards, which is why I'm such a Spice Girls fan. But you can choose when you were in your prime as well. But it does seem to be that those memories, particularly later in life, are the most vivid ones that stick with you. So we're keeping well to time. This is good. I can relax a little bit myself. But it's a great pleasure now to invite Suvankar Pal to come and speak to us about a very innovative, very important trial, MND Smart. And as I mentioned, it's a model that is inspiring lots, a bit like the Spice Girls, you could say, inspiring a lot of copies. And so a lot of other conditions are following a similar model of the platform trial. But over to you, Suvankar. Thank you.
Thank you, Tom. Thanks for the Spice Girls reference. It's great to be here today, and thank you for the invitation. I've spoken at this meeting a couple of times before, so it's particularly good to be back to give you an update on how things have been progressing since we were last together. So just by way of background to set the scene, many of you will know motor neuron disease is a particularly devastating progressive neurological disorder. People present with weakness of their arms and legs, difficulty with speech and swallowing, and ultimately also breathing difficulties. And about half of people living with MND have cognitive changes and behavior change. and about 15% of people actually fulfill the diagnostic criteria for frontotemporal dementia. It's a tough diagnosis to make. It actually takes about a year from when people first develop symptoms to when a definitive diagnosis is made. And thereafter, progression is relentless. So within 2 to 3 years, the majority of people with this condition will die. And unfortunately, 1 in 3 people will die within the first year of diagnosis, usually from complications of respiratory failure. It's not uncommon. So people might think it's an uncommon disorder, but actually the lifetime risk is 1 in 300, which is the same as a condition like multiple sclerosis. And over the last decades, we've understood a lot more about the pathology of the condition. We understand a lot more about the fact that it affects the motor cortex, the spinal tracts, the anterior horn cells, and that in 97% of people, it's due to a misfolded protein called TDP43. And so these allow for us to work towards targeted therapies for the condition. But the reality is that in the UK and globally, the only licensed drug that's available is a drug called Riluzole, and this is a medicine that has been around since 1995. So there's a real urgent unmet need for better therapies to improve outcomes for people living with this condition. And there's been loads of awareness raised by people like Rob Burrow, Ewan MacDonald, who's funded our centre, and Doddy Weir. So there's a huge momentum now around improving access to trials and participation in research for people with MND. And people who live with this condition will tell you that there's nothing worse than living with MND, and they will participate in trials that may work to slow, stop, or reverse this, regardless of side effects, regardless of the burden of taking part in trials. And really, the analogy is drawn with cancer, where it's an extension of clinical care. And it hasn't been through want of trying. So although we only have 1 licensed drug treatment, Riluzole, there have been over 125 trials in the last decade that have tried to answer the problem about identifying better treatments for MND. And the problem with many of these is that they've been poorly designed, so they've been underpowered, haven't followed up enough people for long enough, the outcome measures have been imprecise, and the drug selection has been quite poor. The other big challenge has been actually trying to get people with MND to participate in trials or have access to trials. And in Scotland, for example, before MND Smart launched in 2020, fewer than 10 people had taken part in a trial for motor neuron disease of any sort. So we understood when we designed the study that there was an urgent need for improved participation, but also innovation in how the drugs are selected, and then innovation in how the drugs are tested in the actual clinical trial itself. I thought I'd just, before I talk about MND Smart, talk a little bit about the projects that have actually underpinned the trial itself. And we're here today talking about success in Scotland and celebrating that. And one of the big areas that we've achieved a lot with is the Scottish MND Register. So this is the first global population-based register for people living with MND and was launched in the late 1980s by Charles Warlow and Rob Swingler from Edinburgh. And they understood at that time that by in-depth study of a population of 5.5 million people, with unified health boards and academia working closely together, that there's a real opportunity to understand more about this condition, the epidemiology of it, but also the natural history of the condition. And so we rebooted this twice, actually, once in 2011, then again in 2015. It's now called Care MND. And what we have is a system whereby we can follow people living with MND very closely from their diagnosis all the way through their disease trajectory to death as well. And around 2015, there was a huge amount of interest in motor neuron disease through the Ice Bucket Challenge and through other high-profile individuals raising awareness. And Scottish Government at that time made a decision to double the number of nurses in Scotland on a recurring basis. So we're in a very unique position in Scotland where we have 1 nurse specialist to every 40 people living with MND, and that's unprecedented anywhere globally. And that's what's allowed us really to deliver such a high level of clinical care, but then also now to move on and kick on and deliver research. So we've established through our previous work that we have 99% case ascertainment of people living with MND in Scotland. We have a digital platform that all 14 health boards in Scotland use using a harmonised pro forma benchmarks against the NICE 2016 guidelines. And we have a very rich history of bioresourcing in Scotland. So collecting DNA samples, but serum, plasma, PBMC cells for stem cell generation, and increasingly digital capture as well from people. And there's a very rich culture because of this nurse specialist network now of people donating brain and spinal cord as well and signing up for that in life. So since this launched, we have over 3,000 people who have consented to take part in the process. And we have a high level of case ascertainment and follow-up of these individuals, and also now participation in observational studies. But what people really want to take part in are clinical trials of new drugs. That said, the register has helped us answer a number of important questions over the last decade. The impact of supportive interventions, the impact of multi-morbidity, about the impact of COVID-19 on survival in Scotland. We know a lot more about how many people are living with MND at any one time, 450 to 500 in Scotland. There are about 200 new diagnoses a year, and that has helped with provision of care in the different health boards. We understand a lot more about what drives the different extremes of young onset and late onset and rapid progressors and slow progressors. And we're working hard to deliver new biomarkers as well. But as I said, what people really want are clinical trials. And so we understood back around 2017-18 that we need a wide interdisciplinary team to work together on solving this problem. And so our team includes basic laboratory scientists who specialize in the discovery science side of things, all the way to clinicians, statisticians, medical statisticians from the MRC Clinical Trials Unit at UCL who have delivered disruptive, innovative trials over the last 20 years, and crucially also people living with MND. We heard this morning about the importance of co-production. Really, people living with MND have been at the center of the study design and delivery throughout this process. And one of the innovative approaches that we're taking is a multi-arm, multi-stage approach, a MAMS approach. And just to talk through the rationale for this, if you think about a conventional two-arm study design where you have an active drug compared to a placebo, two-arm trial, by the time you've identified which drug you want to test, gone through writing the protocol, getting the regulatory approvals, getting the funding, training the sites, opening the sites, following up the participants, collecting the data, cleaning the data, analyzing the data, then reporting on the data, all of that will take a minimum of 4 years, a minimum, maybe more, 5, 6. And so if you're going to test 4 drugs in this way, in series, what will happen is that it will take the best part of 2 decades. And after each trial has ended, you lose all of the infrastructure that you've built up. So all of the site teams that have been trained, all of the know-how and the knowledge that you've built up is lost. So it's a very inefficient way of testing medicines. And so the approach that we've taken is the MAMS approach, the multi-arm, multi-stage approach, where we're testing multiple interventions simultaneously against a contemporaneous placebo. And we look at all of the accumulating data that, as we're gathering it, at fixed interim analysis points. So we can look at the data as we're accruing it, and we can look to see, according to pre-specified criteria, whether a drug is working and whether we should invest more time and effort in it and follow up more participants for longer, or whether we should definitively stop that drug. And equally, we can bring in new treatments at any time when the evidence base is there to bring them in. And this approach has worked extremely successfully in cancer medicine and infectious diseases. So there's a trial called the STAMPEDE trial, which has changed the standard of care of prostate cancer on 3 occasions. And many of you will have heard about the COMMEND trial and the recovery trial in COVID-19 that led to acceleration of vaccines for COVID-19. And this approach is not just more efficient in terms of time, but also cost, participant burden. So it's a really neat approach to trying to identify answers to research questions in a more timely way. I mentioned about co-production. Really, as I said before, people living with MND said they want the conversation to change from one of bad news and palliation to one where there is access to research and clinical trials, and much in the way that if people are diagnosed with cancer, there's often a discussion about 2 or 3 different treatment trials that might be running that they're eligible for. The opportunity to participate should therefore be the standard of care. people wanted inclusive trial criteria. So one of the criticisms of historical trials was that if you were young or old, you were excluded. If you were a rapid progressor, you were excluded. If you were a slow progressor, you were excluded. And this was to try and get results from a homogeneous population in a quick way. But that's not really generalizable to the whole population. So people wanted access to trials, and they didn't want to travel past 2 or 3 different hospital sites to be able to access a study. So particularly for people living in Scotland, one of the challenges was that there were really, before MND Smart launched, 5 centres in the UK that would deliver trials. And these were many, many miles away. So people would have to go, travel distance, stay overnight, have the assessments, come back and go backwards and forwards every 2 months, which obviously isn't easy in a condition like motor neuron disease and would lead to high rates of attrition. The other thing is, if possible, could medicines be given in liquid form? We know with people with motor neuron disease that swallowing is affected, and over time it becomes difficult to swallow tablets, and so that might lead to attrition in trials as well. But fundamentally, it was about equity of access. So we implemented a number of things like liquid IMP, couriered to people's homes so they didn't have to come backwards and forwards to collect the drug, video conferencing-based follow-ups, and this was even before COVID-19, and the use of digital technologies increasingly to sort of democratize access, decentralize trials, and lead to more people being assessed in the community. Okay, so this slide summarizes the current study design. We launched in February 2020, and at that point we had 3 arms, a placebo arm and 2 active interventions, memantine and trazodone. So memantine you'll be familiar with is a drug repurposed from dementia, an NMDA receptor antagonist. Trazodone is an antidepressant. Those 3 drugs launched in February 2020, and the primary outcome measures that we're looking at are people's functioning using a scale called the ALS-FRS, and also survival. And I mentioned before, because of the rapidity of disease, unfortunately in MND this is something we can measure as a hard outcome that may not be the case in other neuroprogressive disorders. And so we had stage 1 interim analysis in March 2022, and at that point we looked at data from 50 participants in each treatment arm that had been followed up for 6 months or more. And according to pre-specified criteria, there was enough evidence to continue treating and to continue following up participants to gain more information about these interventions. And so the stage 2 interim analysis occurred last October, and that's when 100 participants had been followed up for 12 months or more in each treatment arm. And at that point, we saw that there was really no difference in functioning or survival between the 2 drugs and the placebo arm, and the decision was made to definitively stop those drugs. And the study was powered to make that decision that those 2 drugs are ineffective in MND. At the same time, we managed to launch our 4th intervention, amantadine, which is a drug repurposed from Parkinson's, in April last year. And because of the infrastructure we set up, at that time, we had 17 sites open in the UK. And once we had regulatory approval for the new protocol, we were able to train all of those sites simultaneously and launch all of those sites on the same day. So I mentioned the efficiencies before with this approach. It means that there's no staggered site opening. And now we have stage 1 interim analysis coming up for that in September of this year. We're looking at a number of other outcomes as well. So we understand that people with MND have cognitive, mood, and quality of life. All of these things are very important. So we're looking at standardized outcome measures for these as well. And we're looking at biomarkers in the blood like neurofilament light chain, which measure axonal damage. So this just summarizes how we've done since we opened. We launched in February 2020, just shortly before the global pandemic. But despite that, we've been recruiting at a steady rate of 15 to 20 participants a month. And we've now opened at 23 sites across the UK. And we're open in all 4 home nations in the UK. And we're delivering the first trials for MND in Wales and Northern Ireland. Really, I mentioned before, we're looking for equity of access. So we're open at big academic centres, but we're also open in satellite district general hospitals. And going back to that adage that we don't want people travelling past multiple sites to have to access a trial. And so we've now randomised just over 800 participants. That milestone was reached last month. And in the 4 years that we've been in operation, we've tested 2 interventions definitively in about half the time it would have taken if we'd tested them sequentially. So the data I mentioned from the interim analysis has been submitted for peer review, And you can see in the first, which one's the point? You can see in the first chart here, this is the rate of change of ALS-FRS, which is the functioning rating scale, and the second graph looks at survival. Essentially, the take-home message is that there's no separation between the three curves for either of those charts. And this was following up several hundred participants for quite a long duration of time, so a really definitive result. But in addition to the hard outcomes that we're measuring in the trial, there are softer outcomes that are important for people living with the condition as well. And they've repeatedly fed back to us that the conversation now has changed. We now have access to trials because of the study across the UK, and this has led to a change in conversation from one of end-of-life care to one of hope, and people understanding that they're contributing to something that may not help themselves today, but will certainly help for people with this condition in the longer term. We've continued to try and innovate as the study's gone on, so we've developed interventions like remote consenting using DocuSign, electronic diary cards that are remote and completed by participants, and remote outcome measures as well. So it's really accelerated decentralized trials and following people up in the community. So I mentioned before that before we launched, there were fewer than 10 people in Scotland who'd ever taken part in a trial. And again, this is part of celebrating research success in Scotland. We've now developed a clinical trials hub at the Ewan MacDonald Centre in Edinburgh that has randomised over 160 participants. And we have nurse specialists, research practitioners, clinical research fellows, all dedicated towards delivering trials for people with MND. And it's not just in Edinburgh, so Dundee was the second site to open in the UK, and it's great to be here today. Tiffany's one of the neuroprogressive support officers who has contributed with Ian Morrison, and they have recently celebrated the randomisation of their 50th participant in Dundee. And Scotland as a whole has contributed immensely, so 303 participants from the 800 have been randomised in Scotland across 5 sites. So it's really an incredible contribution from Scotland. And we heard this morning, I think Tom at the very start of the day was talking about access to remote areas. So the Aberdeen team has delivered an example of this where they have managed to randomise participants as far remote as Orkney and Shetland, where really people haven't taken part in trials of any disorder before. And this has been possible due to really innovative thinking by the local trial teams and local understandings. They've used offshore companies that provide couriering of IMP to people's homes. The local teams are very well set up in telehealth. We've used DocuSign, as I mentioned, for signing of documents and reconsenting. And when the local clinicians are visiting the islands for NHS visits, we've managed to tie in the research assessments at the same time as well. I mentioned that Wales is a site in Cardiff, so they hadn't had any trials for motor neuron disease before, and really they've very quickly randomised 50 participants there. Similarly in Northern Ireland, this has been part of setting up a wider clinical research network for MND studies. And Rayburn Forbes, who's the academic neurologist leading the study from Belfast, was actually the research registrar who set up the research register for motor neuron disease in the late 1980s, early 90s. So it's great for him to be kind of fulfilling a cycle and now delivering trials for people in Northern Ireland. We have a number of hub centres throughout the UK that I've mentioned. Some of the larger ones like Birmingham have a very ethnically diverse population, which leads to generalisability of the trial results, which is great. Similarly for Newcastle, and both of these have recruited 50 participants recently. And the messages that come through from the sites is that it's changed the conversation for clinicians and researchers as well, where they feel like there's more hope and optimism and that they're delivering something that is productive and has changed their working day. So the team for MND Smart now includes over 150 people from our 23 sites, and this ranges from drug discovery scientists all the way through to the clinicians delivering the study. And this is a picture from a sunny gathering that we had in Edinburgh last June. There was about two minutes where it stopped raining where we could go out and take the picture. So really, we're always grounded and motivated by our PPIE group, and they're always telling us to keep moving forwards, keep innovating, and not to rest. So The plan to introduce new drugs opens up a new avenue of hope, and unless we try new things, we're stuck in the same place. So, tested 2 drugs, what's coming next? Well, we heard about drug discovery in the previous talk. It's really an exciting era of drug discovery in motor neuron disease because there are a number of different evidence-based approaches that are converging together to select medicines. And one of the approaches we're taking at Edinburgh is through high-throughput stem cell screening. So we have very sophisticated stem cell-based models of the disease from human stem cells grown in a dish that can model what the disease is like in humans. So we have a model of the misfolded protein TDP43 aggregation. We have models of nerve excitotoxicity and also models of the supporting cells, the astrocytes and microglia. And we can screen thousands of drugs at pace using robotic methods for stem cell screening. We also have an active group that's doing a large structured systematic review of all the literature in neurodegenerative diseases across other conditions that share common pathways with MND, things like Parkinson's and progressive MS and dementia. And there are a number of sophisticated animal models like TDP43 mouse models and zebrafish models that are emerging. And so all of these lines of evidence produce a long list of drugs that our expert committee can look at and consider the practicality, the feasibility of bringing these through to clinical trials. And so an example of this are 2 drugs called tacrolimus and baricitinib, which we're working up protocols for to introduce in the trial next. Tacrolimus is a drug that's repurposed from transplant medicine that's used as an immunosuppressive. But at low doses in our stem cell assays, it has reduced TDP43 aggregation and dampened astrocyte reactivity. And so we're working now on considering how we can deliver this to people with MND. And the things that we need to consider are safety monitoring, the route of administration, dosing schedules. And similarly, baricitinib is a drug used for rheumatoid arthritis, but there's a compelling evidence that it would work from the stem cell assays in MND as well. And one strategy for the longer term, which has worked really well in infectious diseases and cancer, is to think about combination therapies that are targeting different pathways. And for a complex disease like MND, this has to be something that should be tried and will obviously lead to more complexity in the trial methodology, but something that we're prepared to work towards tackling. So in addition to the clinical trial outcome measures, the platform has provided a huge opportunity for reverse translation. And so what I mean by that is that we've got now 800 participants worth of information over a long period of time with a high level of clinical annotation at 2 monthly intervals. But we also have a bioresource where we've been collecting blood samples at 6 monthly intervals and increasingly also aspirations for collecting digital data. And so we've been collaborating with the UK Dementia Research Institute, Biomarker Factory, and colleagues internationally to work on these samples that have been collected at 6 monthly intervals. We're working with the MRC phosphorylation unit here in Dundee and also with Gates Ventures, an international collaborative group. And here we're looking at whether we can look at developing biomarkers for early detection. I mentioned at the very start, it's a very difficult diagnosis to make. So can we have a diagnostic assay from this? And can we also have better markers of disease progression, so better secondary outcome measures for clinical trials? And we're looking at high sensitivity immunoassays like Simoa and Neulisa as a method for doing this that can just look at small amounts of blood collected from participants and look at a whole panel of neurodegenerative markers. Voice is something we're very interested about, and just as a brief aside, we have launched a digital Pathfinder project in collaboration with industry earlier this year, and the aim of this is to collect voices from multiple cohorts of people living with MS, Parkinson's, dementia, and motor neuron disease, and again, from the acoustic signatures in people's voices, see if we can detect early markers of diagnosis and a marker that would be surrogates in clinical trials and for monitoring of people's progress. And where do we sit in the wider landscape of multi-arm, multi-stage trials in neurological conditions? Well, we've set up the Accord collaboration about 3 years ago now, and this is anchored at the MRC Clinical Trials Unit at UCL. And there are a number of other trials that using the multi-arm, multi-stage approach now. The Octopus trial, led by Jeremy Chataway, launched last year. And the ACT PD trial for Parkinson's disease will be launching later this year. Paresh Malhotra, who's lurking in the corner of this picture, is speaking tomorrow about the Mastodon trial that's launching, or that's being protocolized now for launch hopefully next year. And the Accord collaboration has a Fellows Academy, so we have a number of young researchers, clinicians and non-clinicians who are interested in trials methodology, drug selection, outcome measures, and we're lucky to chair a group of 20 fellows now across 15 different university sites across the UK, bringing on the next generation of clinical trialists in this area. So all of this work really wouldn't be possible without close collaboration with funders. And it's a different way of funding studies. We're talking about infrastructural projects that last for many years. And so we've been very fortunate to have a close relationship with MND Scotland, who are one of our foundational funders, and Jane Haley, you'll be hearing from next, has been working closely with us. And we've now secured funding really till 2028, thanks to close collaboration with MND Scotland, MNDA, the Alan Davidson Foundation, the Doddy Foundation, and also LifeArc. So in terms of forward momentum, we're really looking to introduce new drugs as soon as the evidence base for those becomes available. I mentioned about combination therapies, and also really thinking about how we might collaborate with pharma rather than just having repurposed medicines if novel compounds come out and they might be suitable for phase 3 testing in this platform. So to summarize, MND Smart with its innovative multi-arm, multi-stage approach has now tested 2 separate interventions in just under 4 years since we launched. And so we've demonstrated that it is a highly efficient platform in terms of time, participants, and financial resource. We've also, crucially, in the last 5 years, just delivered infrastructure that didn't exist before. Only 8 of our 23 sites had delivered clinical trials for MND before. And now we have 23 very research active sites. You've seen the recruitment numbers across them. Working closely with our PPIE group has been really important. We're often conveying complex messages about the trial design, and they've been crucial in helping us with all of the patient-facing information. But also when we come to decision points about whether a drug should stop or continue, with the communication plans that we share with people. So they've been crucial in that as well. And I mentioned about the bioresource being really critical in forwarding our understanding of biomarkers in this condition. So none of this would be possible without many, many people across the country. We've got our key collaborators in medical statistics, the site teams across the country, various trial committees, and really we've been kind of building the plane and flying it at the same time, and it's thanks to all of these people that we've been iterating the protocols and making sure things run smoothly. But crucially, the people to thank are the participants and their families and our PPIE group. So I think I'll stop there, but you can follow our updates on social media and through our website if you're interested. Thanks very much for your time. Thank you.
Thank you very much, Suvankar. I'm sure there must be some questions. So firstly in the room, but also we wonder if there are any online.
Thank you for your talk, and obviously the MND Smart is just an amazing design in itself. I noticed you have a placebo group that you use to compare against the 2 drugs, and then when those 2 drugs finish, you use another 2 drugs. Do you use the same placebo cohort for the first 2 drugs, and then do you use that cohort, the same cohort again for the next 2 drugs, or do you have to get a new cohort of placebo as you're going throughout the trial?
Yeah, thank you. So it's always contemporaneous placebo. So the participants in the placebo arm are receiving the placebo always at the same time as the people receiving the intervention. There's a lot of work being done in MND trials about whether you can use historical placebo data or synthetic placebo model data. At the moment, there are some fallacies with that approach, and I think for the regulators to definitively know if a drug is working or not, we still need that contemporaneous arm.
Yeah. Are there any questions inside it?
Yeah, there is. Okay, I apologise for my ignorance in how I pronounce this, but I don't know what this is. So how is astrocytic reactivity implied in MND?
Yes, the supporting cells are the astrocytes, and they are involved in MND as well as the neurons as well. And so the astrocytes are overactive, really. And so if drugs can dampen astrocyte reactivity, which some of the models have shown, they might be helpful in MND, particularly in this combination approach of different drugs targeting different pathways.
Great, lovely. Well, that's about past, so I think maybe we'll stop there if that's okay.
Thank you.
So join me again in thanking you, Suvankar, for speaking, please. And we now have a half an hour break for more looking at posters, looking at the stalls, drinking teas and coffees. It looks like there are things we probably shouldn't be eating that we can choose to eat if we want to. And that if you could promptly come back here at 3 o'clock for the next session, that would be very much appreciated. Thank you.
When I wake up, well, I know I'm gonna be the man who wakes up next to you. When I go out, yeah, I know I'm gonna be the man who goes along with you. If I get drunk,